Basically, there are various structures involved in the process of excretory system of a cockroach. Basically, if you take the process of excretion in Periplanet americana, it is basically occurs by specialized structures like Malpighian tubules. As you have already learned about Malpighian tubules in the previous classes. Now, how these Malpighian tubules looks like? Let me draw the structure of Malpighian tubules so that you will understand how they perform the excretion process. All these Malpighian tubules are directly opening into the hind gut of the cockroach. Now basically these are lined with specifically these are lined with specialized cells, Now this is the secretory part of the Malpighian tubule, and whereas this is an absorptive part of the Malpighian tubule. Now let us see how the excretion is performed. Excretory system of a cockroach helps in eliminating nitrogenous waste from the body. Now here it helps to eliminate basically uric acid. So Periplanet americana is considered as a uricotelic animal. Apart from this, the excretory structures are such as like Malpighian tubules. What are those tubules? In the first slide I have drawn that. Fat bodies, uricose glands and nephrocytes and cuticle are important excretory structures of the cockroach. Now, long and unbranched yellow colored tubular structures which are arranged in 6 bundles. They are arranged in 6 bundles. Each bundle contain 15 to 20 tubules. These 6 bundles together approximately 100 to 150 yellowish colored tubules are present, called as Malpighian tubules. Now, a great scientist called Marcello Malpighi, you know very well about Marcello Malpighi, called them as vasa varicosa, and here Meckel coined the term Malpighian tubules. The great scientist Meckel coined the term Malpighian tubules. Now, apart from this, if you carefully see, each tubule is lined by single layer of epithelium on the inner side, and the distal part, if you see, the distal part of the Malpighian tubule is secretory and anterior part is absorptive. Now, glandular cells of Malpighian tubules basically absorbs water, salts, CO2 and nitrogenous waste from the hemolymph. Now, cells of proximal part of tubule. The proximal part of tubule reabsorbs more water and some inorganic substances like inorganic salts. Now, by the contraction of tubule, urine is pushed into the gut called ileum. Now, more water is reabsorbed from it, hence it is necessary to form the solid uric acid from the fecal matter. Now, reabsorption of water from the waste and formation of dry uric acid in the fecal matter or in the fecal waste is one of the method which helps in the process of conservation of water in terrestrial animals. Fat body is a lobed structure. It contains urate cells present in these bodies associated with excretion in a way. Now, these cells absorb uric acid basically throughout the life. Now, it is a phenomena called storage excretion, as they maintained stored in the urate cells for longer period of time in the corpora adiposa structure. Uric acid is stored in the uricose glands. These uricose glands basically present in utriculi majoris, in the mushroom shaped gland of the male cockroach. Male cockroach contain a mushroom shaped gland which plays an important role in the uric acid storation, act as a uricose gland. Now finally, cuticle, that is an outermost layer of the cockroach, also plays an important role in the process of excretion. This cuticle helps to eliminate the uric acid during the process of molding. You know, molding is a process, or it is a kind of metamorphosis during the development of a cockroach. Because of this molding, little amount of uric acid will be eliminated. This is how excretion is performed in the excretory system.